Okay, it's after 3, so I think we'll start this final PhD completion seminar of Marek. Marek is unique in that I think he's got the most supervisors of any student I've ever seen, and in fact, what's interesting is about half of them are actually here. So Marek's interesting in that he's tried to apply bioinformatics to clinical cancer evolution problems, and he realised quite early on in his PhD that one of the issues is that no one's looking at structural variants for the evolution of cancer. So Marek's developed a method which he's going to go through in some detail today about how he's used structural variants to call evolution in cancer.
Thanks for the introduction, Chris. I would just like to begin by acknowledging the traditional owners of the land on which we meet today, Wurundjeri people of the Kulin Nation, and I pay my respects to elders past, present and future. So the prevailing theory of cancer evolution is that cancer begins with a single progenitor cell that has acquired mutations that turn it into a cancerous state. And as it acquires mutations, it divides and grows, and the tumour mass grows, and this leads to a differentiation of different clones that have similar genotypes. So when we refer to a clone, this is a set of tumour population cells that have similarities in their genotype. And as in Darwinian natural selection, you can have this kind of branching process where you have different lineages that differentiate from each other, and they can give rise to more clones that are further lineages from there on. So in this example, we've got clones B and C that have evolved different genotypes, and in this, and they've all come from this ancestral clone A. And at this point in time, there's a therapy that has been introduced in this particular patient tumour, which leads to shrinkage of the tumour mass and this extinction of clone B. However, clone C has some particular mutations that allow it to survive this treatment. So it goes through this population bottleneck, and then you've got this relapse occurring here where the mutations allow it to become resistant and then allow it to resume its unrestrained growth. And then you've got this clone D that arises as an ancestor from clone C. The biopsy, if you take a biopsy at this particular point in time and you have a bulk tissue sample, and you could, if you look at the different mutations that are present within these clones, you might be able to differentiate that there's this one large clone B that it's descended from this clone A as well, and then you've got this clone C that is defined by unique mutations. If you also took a biopsy at this point in time after the treatment, you would also be able to look at the different mutations and then see, okay, we don't have any of the unique mutations that we saw in clone B, and therefore this clone has likely become extinct. But you've got these new mutations that have arisen that we can correlate with clone D here. So my talk will be delivered in 2 parts. The first part will be about the method development, and the second part will be about application. So to give you a brief overview of my thesis contribution, I've developed a method to study clonal evolution of genome rearrangements, and I'll talk about all the details involved in that. Then we applied this method to over 2,700 whole genome pan-cancer samples with over 37 different types. And then we developed a strategy to test for evidence of clonal changes in a small prostate cancer cohort where there were some low-quality samples, and we wanted to look for differences in clonality longitudinally. And then that's how we applied these methods as well. So this is the first part of the talk. I'll be talking about svclone, which is the method that I've developed. So as I was talking about the mutations that you can correlate with different clones, different cancer types have different enrichment of different kinds of mutation. So this is a pan-cancer analysis. So for instance, you can't just use one type of approach when you're looking at the clonality, because you're going to have different types of mutational enrichment across different cancer types. And this is just an example of it. So this is a paper from a few years ago where they looked at 12 different cancer types. And then they were able to separate these different types into two different subtypes. So high levels of recurrent single nucleotide variants and then high levels of copy number aberrations that were recurrent. At the sort of high-end SNVs, you've got some kidney cancers, colorectal adenomas, for instance, and then on this side, you've got the ovarian and the breast cancers and also lung squamous cell. And then you had some cancers that had intermediate levels of both, such as lung adenomas, and then so you can see that you can't just use one approach to study different tumor types. You have to be aware of what are some of the recurrent mutations across cancers. So as a high-level overview, if you were studying clonality and changes across time, or also correlating your different genotypes from your clones to particular clones, then you would be looking at getting a whole bulk tumor sample and also a germline sample and then running some sequencing. So this could be whole genome sequencing, it could be whole exome. And then you would do some variant calling such as SNVs, indels, somatic copy number variations, and structural variants. You would then use some kind of clonal deconvolution methods in order to associate your mutations to your clones or figure out how many clones there are. And there are many different methods available if you want to use an SNV-based approach. For instance, PyClone is one particular approach that is used quite often. If you want to look at the clonality of your copy number segments, there are quite a few tools available. However, it's much fewer than SNVs. One of them is Battenberg, for example, where you can segment your different copy number segments and figure out which ones are subclonal and which ones are clonal. Generally, you will get a lower resolution with copy number segments, as you might not be able to distinguish as many clones, and because it's more difficult to do with copy numbers. However, if you're looking at structural variation, particularly balanced rearrangements, so let's say you've got this EML4-ALK fusion that you found in your sample, and then you know by running your heterogeneity analysis that you've got some clonal mutations such as TP53 missense, and then you've got EGFR amplification, which you've correlated with clone B. However, because this fusion is caused by balanced rearrangements, you really don't have any methods available in order to figure out which clone this comes from. And this could be important in the context of finding particular biomarkers or looking at particular mutations that might give resistance. So at the moment, there's not much available here, and this is the gap that we're trying to fill. So just to make sure that everyone understands the term structural variation, these are large genomic rearrangements that occur across large stretches of DNA. Copy number variations are a subtype of structural variants, and you've got, so for instance, this could be a large segment in the genome that gets deleted. It could be a segment that gets duplicated, either in tandem or interspersed into a different part of the same chromosome. But then you can also have rearrangements that don't affect the total copy number of your chromosomes, such as inversions, where a segment just inverts on itself, or one part that gets cut out and then translocated somewhere else, or an interchromosomal translocation, where one part becomes fused to another chromosome. This could be whole arm events, or it could just be a chunk. So these are termed balanced rearrangements, and this is what we're interested in terms of using this methodology, even though you can also use this methodology for copy number variation. So what is a variant allele frequency? So in order to calculate the cancer cell fraction, so the average proportion of cells that a particular mutation is in, and then also your clones will have a cancer cell fraction, you need to start with something called a variant allele frequency. So in the most simple terms, here are your reads stacking up onto your reference genome. So this is your reference genome here. You've got 10 reads, and let's say you're interested in this particular base pair where the reference is A, and then you've got 6 A's here that align with the reference, but then you've got 4 G's, and you've filtered out any germline artifacts, so you're pretty sure that this is somatic. Well, in order to calculate your variant allele frequency, you would take your variant bases, so 4, and then divide it by the total number of reads. So your VAF is 40%. So that's quite simple, and that's what makes SNVs very nice for doing this kind of analysis. However, structural variants present some more complications. So these are your breakpoint boundaries, and these are your whole genome sequencing reads that are aligning at this particular point. So let's say that there was a deletion at this point, so this has been cut out in the tumor genome. And if you had, say you've got one read that is crossing the boundary and it's affected by this variant, you've got this segment which is split at this point. So these base pairs actually align on the other side, and these base pairs align over here. So this is something that's called soft clip in your aligner, and you're going to want to use that as evidence for your structural variant. So what you want to identify are the supporting reads and also the reads that aren't supporting the variant. So you're always going to have some normal contamination. So these are your normal reads. These could be on another allele, which doesn't contain the variant, or they could be just part of the normal tissue. And then lastly, you've got spanning reads. So spanning reads, when you run whole genome sequencing and you've got paired end reads, you've got these kind of pairs of reads, and you don't know what the intervening sequence is, but you know roughly how far they are apart. So when there's a variant in where you've got spanning reads, one on each side of the junction, that means that this intervening sequence is a different size to what you expect. So because this is cut out in the tumor, that means that the size is actually much smaller than is shown here. And you can use this information in order to classify these reads as tumor reads, even though they don't actually cross the boundary. Now you can also have a spanning read that has a bit of a soft clip, that's fine. And then these reads, the other read is actually over here, and that read is actually over there. So these are a few criteria that we use. And this is what it actually looks like if you've got your IGV genome browser. This is like a histogram. And then the variant is occurring here. So you've got a deletion occurring at this point. And these are the soft clips. And these are the normal reads where they pass the boundary, but they're not affected. These are split. And then you've got the discordant reads here. So that means that these reads are spanning reads where the insert distance is not what you expect it to be. And then you can use that information. It has colored it red because the insert distance is different to what it expects. So we utilize this in a read counting approach, which uses a couple of heuristics. So we have this soft clip boundary that we say, okay, a split read has to pass this boundary by some X number of base pairs. Is usually 10 by default in our program. And then also we apply that same criteria to the normal reads. So they have to pass this boundary so that we're counting equally. So there are a number of different other criteria. So let's say split reads, they have to cross the break. They have to be past this threshold. And then they have to have a valid insert size. So valid insert size is something we want on every read. So you have an insert size distribution, and you're going to have a mean of that, and we want it to be within the mean plus 3 times the standard deviation. And of course, when you've got spanning reads, you want to adjust for that insert distance. So you count from here to here and here to here, and then you use that as your modified insert distance. Another extra parameter is that we make sure that the spanning reads are facing the break. So each read will have a directionality, and the spanning reads have to face the break. And so you'll also notice that we get 2 variant allele frequencies, one at each break end, and this is because of the different number of normal reads. So we can just sum the split reads and then also the spanning reads, but however the normal reads will be different at each locus. So I'll mention that a little bit later, but we essentially just use the minimum in order to decrease our search space when we're doing our clustering. However, we also go back and adjust that later. With doing some simulations, we encountered a few different artifacts or biases that we need to correct for. So when you have a duplication, you've got your reads that are duplicating, and then you've got the reads that will pick up as being variant reads. So if you've got a sentence and then you copy, say, half that sentence and paste it, well, you've got half a complete sentence, so that's your structural variant, but you've still got your original sentence. Meanwhile, if you took a sentence and then you swap some letters or you cut out a piece, then you don't have your original sentence. So we need to adjust our normal read counts. The other rearrangements basically get rid of the normal reads that would be there if it's happening on that particular allele, and then you've got no normal adjustment that's required for them. What all other rearrangements, we need to adjust the supporting reads just slightly because of alignment issues. So for instance, we tried this with Bowtie, and it's a little bit better at adjusting for the splice junctions. So we were able to use a lower factor, but we just use a linear scaling factor. For instance, with BWA, you need to adjust a little bit more because it's a little bit worse at mapping those reads properly. So this is what it looks like when we have a simulation and we adjust the VAFs. So this is a 100% purity sample, and we expect the VAF to be around 50%, so occurring on 1 out of 2 alleles. So this is just one simulated chromosome. So we can see that the deletions, the adjusted is the green, so we just have to slightly move deletions, insertions, and translocations with the supporting read, just to slightly adjust them. But duplications require a larger adjustment, and you can see that there. So this adjustment factor also works on purity and ploidy, so it kind of dynamically scales to your sample. This is just a large zoomed out outline of the SV clone pipeline. So I've talked a little bit about counting reads. In order to do this counting step, you need to annotate a few features, such as the direction that the SV is facing. If you don't already know this, if your SV caller gives this to you, then that's great. And then we also classify the events based on the direction as well. And there's a couple of heuristic rules we use for that. So you need your SV calls and your corresponding BAM file. And then you can run this pipeline. And then filtering, we use some quite strict filtering criteria in order to minimize the artifacts that you get in your clustering, because you're going to have far fewer SVs than you are SNVs, just the nature of these events. Optionally, you can also add your SNV events. This is going to allow you to do some sanity checking, or if you've only got like 5 SVs, but you've got a couple of hundreds or thousands of SNVs, you can cluster on those and then just see where your SVs lie. The clustering algorithm will treat them the same. And then you can also attach your copy number states. So I'll talk about this, why we need to adjust for copy number, because it's quite important. And then in the end, if you filtered out some variants that you want to add back in, you can run the post-assign script. In the end, you'll get the clonality of each SV, the number of clusters there are, and then you can also match this with copy number and run some other analyses on it if you like. So I mentioned this term cancer cell fraction a few times. What is cancer cell fraction? Well, this is your bulk sample. You've got normal cells. This is part of your normal contamination. That just are almost always present in your bulk tumor. Then you've got a couple of different clones. So you've got clone A here, and then you've got clone B present in slightly different proportions. Well, to calculate CCF, we want to get rid of the normal component. So the cancer cell fraction of this particular clone is 2 out of 6. It's just a proportion, 33%, and clone B is present at 66%. However, it's not just this simple, because you can't infer whole genotypes just based on the cell types. There's no way to do this. What you have to do is you have to infer the CCFs from groups of variants that have similar CCF. Copy number is a factor that confounds this kind of analysis. So let's say you've got a structural variant here on one of these alleles. You've got a normal population at copy number 2, because these are diploid cells. So you're almost always going to have copy number 2 unless in males on the X chromosome. And then you've got this tumor reference population. Let's say there's been 1 allelic duplication, so you've got copy number of 3. And then you've got tumor variant population where there's been potentially a whole genome duplication, and then the variant has occurred afterwards on only 1 of the alleles. So if you did a VAF calculation of this, you would get around 11%, because this variant is on quite very few copies relatively, but its true CCF is still the population CCF at 66%. So each variant CCF should correspond to the fraction of the tumor cells that contain that variant. in order to go from VAF to CCF, here's your variant allele frequency, and then we perform some kind of transformation to get our cancer cell fraction. So unfortunately, this slide contains some maths. So hopefully you'll be gracious enough to grant me one slide with maths. So the probability of sampling a variant read is something that we can define in a binomial distribution. And this Bj is the number of variant reads. Dj is the total depth at each particular locus, so each locus j, each corresponding to a breakpoint. And the Pj is that probability. So this is the formula we use to transform from VAF to CCF. We can break this down into a few different components. Here's the normal component. You've got this pi rho, which is the tumor purity, and then you've also got the total copy number of the normal, so this should almost always be 2. And then you've got your tumor reference. So this contains a few other parameters. You've got your tumor reference copy number state, and this you will obtain from a copy number calling algorithm that lets you look at clonality. So that's where your fraction comes from, and you've also got your copy number as well. And then you've got your tumor variant population. So you've got your the fraction at which it appears, and then the copy number, and then also the purity. And this is the fraction of alleles that the variant occurs on. So you can get a PJ for each variant. And one thing I've left out is the phi j. So phi j will correspond to the cancer cell fraction of the tumor population. So this is what you want to infer from all of that. And this is the way that we do this. So you can calculate this in a Bayesian way. However, to calculate posteriors is very mathematically very intensive. You can't really do this on current computation because it requires multi-dimensional integration. So one way to do this is to use Markov chain Monte Carlo for this. So we define distributions for our unknown parameters. So anything that's in white, we don't know. So these are the copy numbers. This is the tumor population. This is just a prior. This is the category cluster that it gets assigned to. But we know all this, right? So that's just what I've mentioned. And then we can generate a number of clusters dynamically. So we use something called Dirichlet process. And then we run this through an iterative approach in order to approximate what the posteriors are. So we know what our distributions are. We have particular data that we want to simulate. So we want to simulate what each one of our supporting read and depth distributions. We can simulate some distributions and pick out the parameters that fit that quite well. And we do this through an iterative approach. So for instance, here you've got your Markov chain, and it's kind of going through random iterations. And if it hits a high probability state, it will stay there. Otherwise, it will keep moving. And through this convergence, we're able to plug all of these parameters into this machine, and then in the end, just pull out the ones that correspond quite well to the underlying distribution that we've got here. So now we can kind of go from the VAF to the CCF. Here we've got our CCF distribution. Here we've been able to cluster it and find the CCF means for each of them. Then we can find out the clonal makeup. So we can say, all right, this is 70% clone B, 20% clone C, and then the rest are at CCF1. So these are your clonal population, and all of the cells contain variants from this population because it's the ancestral population. And then this could correspond to a number of different phylogenies. You probably wouldn't know unless you had multiple samples or you had samples further up this phylogeny. We could say, based on these proportions, this could be a branching evolution event, or it could be linear evolution where clone A has given rise to clone B, has given rise to clone C. So phylogeny is not a focus of our algorithm, but it is something that you can infer, and it's good to keep in mind because some of these algorithms will let you do it. So we've made our tool quite user-friendly and made sure that we get a lot of information that the user can use to figure out what the best fit is for their data. So always you're going to be wanting to run a number of different runs for each clustering attempt. So here, for instance, we've got, you can see how the data has been segmented for each different run, and then you can see a summary of the clustering where you can see the CCF means and then also your corresponding variant CCFs. And then we also have a fit metric, so in order to figure out what is the best fit, so anything that's the lower the fit metric is, the better the fit. And then you can look at your SVs in a Circos plot, correlate them with structural variance, look at the SNV density based on the clonality, and then also get some really robust output for each run. A lot of the time, just doing a visual analysis to see how well the clustering has performed will give you an idea about whether it's a good fit or not. In order to actually validate this, we used a real sample. So in this field, there tends to be a lot of simulation that occurs and people simulating very clean-looking data sets and then running their algorithm on it and then getting great results. But we wanted to push it a little bit further. So we had this, from a previous study, we had a patient that had 2 metastases that were diverged from a common primary clone. So they had shared mutations, but they also had a unique mutation, so private mutations. So they had similar coverage and purity, and what we were able to do is take these 2 different METs and subsample them in different proportions and then merge them together. And doing that in different proportions, such as 90-10, 80-20, we should be able to get out these respective proportions and then also 100% for the shared mutations that are present at this branch A. These are some of the results from this. It's not all of the results, but it's a representative amount. So this is the best run of the SV clone. You've got your histogram of cancer cell fractions here, and the blue lines are the cluster means. So here it's been able to segment these clusters into, it's been able to find 3 different clusters and segment them into different CCF means. Here is the optimal distribution. This just means we didn't run clustering, but we looked at the ground truth of the BM and GM. So was the variant in a particular MET only, or was there evidence that they were in both METs? And then used that as our sort of truth metric, and then just ran it through our CCF calculation formula. So you can see that the underlying truth is actually pretty messy, and you've got some clusters that are overlapping and don't correspond quite as well. We think that this is just a nature of the data. It's very noisy and it's very difficult to get very clean frequencies from this. If you contrast that to the SNVs, you can see that these are the optimal distributions. You can see that you get quite clean distributions here, and that's largely due to the more samples, more data points that you get from SNVs. There are about 5,000 here, whereas this is 142, 200. So in order to figure out if we were getting good results compared to another clustering algorithm, so PyClone is a state-of-the-art SNV clustering algorithm. The size of these circles just indicate the CCF mean that both of these algorithms have found, and this is the truth. And then the size indicates the amount of variance that were assigned to that particular cluster. So you can see you get pretty good concordance in some of these clustering situations. We found in SVClone, we found 3 clusters across all of these, and also in all the other mixes that we found as the best solution, which is good, as there were 3 underlying clusters. PyClone sometimes missed one of the clusters, but its CCF means tended to be a little bit more accurate. So there were pros and cons to using both, but on average they were comparable. So in order to close this gap that we mentioned earlier, you've got your once you've sequenced your sample and you've got your ITH analysis, then you can look at your SVs and then find out the clonality to figure out that, okay, this EML4 ALK fusion actually is likely to be in clone C. So just a summary for the methods component. We've developed a pipeline for inferring the cancer cell fraction from structural variants, and we use these rule-based read counting methods, and also clustering using Bayesian inference approach. And then we validated this using simulations on the VAFs and also real sample mixtures as well. So this is the only method that allows us to consider balanced rearrangements and provides this deconvolution of breakpoint data, no matter what the structural variant is. So in order to apply our algorithm to a really large cohort, we are part of the Tumor Heterogeneity and Evolution Working Group in the Pan-Cancer Analysis of Whole Genomes project. And so we applied this algorithm to those samples, and then also an aggressive prostate cancer cohort, which was smaller scale and also a more specific research question. So the PCOG is an international project with over 2,800 donors, over 2,600 tumors, 801 terabytes, so lots and lots of data. It's a huge project, and we're just a very small part of it. So the amount of computational effort that actually had to be undertaken to just get this plot is a lot. So we need to get the consensus single nucleotide variant calling, and also the consensus SVs and the consensus copy numbers from a whole bunch of different groups around the world, and then run our algorithm across over 2,700 different samples, which took weeks and weeks, especially with having to iterate and finding the right approach and ironing out the bugs of doing that. So the effort was immense, but we managed to find some really interesting things. So this is a number of different histology types separated by mainly primary organ. And on the x-axis, you've got the fraction of subclonal SNVs. So each data point is one patient sample. And we simply looked at whether the CCF of a particular variant was below 0.9. If it was, we called it subclonal, otherwise we called it clonal. So each point just kind of corresponds to that for SNVs, and then also for SVs. And this was all run using SVClone, SNVs and SVs, in a sort of co-clustering. So there were some interesting things that we found. So for instance, in breast cancer, you can see you've got a bit of an enrichment of subclonal SVs compared to SNVs. That's also the case with pancreatic and ovarian. And what we were quite interested in, however, was we noticed this cohort of liver cancer samples that had high subclonal SVs and low subclonal SNVs. So digging around a little bit more, we managed to find this subclonal neutral rearrangement phenotype, where you've got an enrichment of so interchromosomal translocations and inversions, so balanced rearrangements in subclones. And this is just an example of that compared to the clone. And then when we looked at the survival analysis, we found that it was much worse than the control, especially even when we stratified it by different histological types, to make sure that we weren't just classifying the more aggressive cancers or anything like that. And we also stratified it by different numbers of SVs as well. So you can see how using SVClone, you can address potentially some clinical questions as well, such as if you're monitoring relapse and you know the particular mutations that you're looking for, you can trace the frequency of a variance over time. So you can already do this with SNVs and maybe even copy number changes. However, if you've got balanced rearrangements that you find that correspond with relapse or correspond with monitoring particular metastases, now you'll be able to use this approach in order to track the fraction. So the final part of my talk, I'll be talking about neoadjuvant prostate cancer cohort. So this was a small group of 14 high-risk prostate cancer patients that underwent neoadjuvant supercastration therapy. So prostate cancer is driven by androgens a lot of the time. So if a form of therapy is to try and reduce the androgens in the body. So you do this through this kind of castration approach. And so biopsies were obtained prior to and post-treatment. So neoadjuvant treatment is usually done prior to surgery, so in order to shrink the tumor mass, and then you do prostatectomy, which was the case in this sample. All of the samples were formalin fixed, paraffin embedded. This is very common in a surgical setting because often you don't have the facilities in order to have fresh frozen samples, so you need the liquid nitrogen on hand, you need to have the storage facilities available. So usually FFP fixation is used because it keeps the tissue at room temperature. However, there are some issues with variant calling as it creates chemical changes to the tissue. So we had 27 samples that underwent whole genome sequencing. The average coverage was around 38x, so this is kind of the low to moderate end. The question was, can we find any evidence between pre and the post in terms of clonal changes? So this is a summary of the cohort. You can see these are the pre-samples and they're all FFPE. We don't have pre's for all of them, and although we also have fresh frozen and FFPE for some of the post-treatment samples. So the idea was to get the samples of different fixation types at the same time so that we can figure out if there are any particular artifacts that are occurring and adjust for them accordingly. So most of the patients had a partial response, 4 of them had no response, and 1 patient had a good response. There are a few more patients in this cohort, but they didn't have usable samples for this analysis. So our first port of call after we did some QC is to run some copy number calling algorithms in order to obtain the purity. So we did this using ASCAT. And for some of them, ASCAT wasn't able to estimate what the purity was. This was probably because of lack of copy number segments, as some of them had also shrinkage of tumor mass, and it was difficult to get the purity. So they could have been completely normal. Although for some of them we were able to get fairly decent estimates, but however we'll show you later on that not having good purity estimates makes things quite difficult. And we also had pathology estimates, however, they didn't really correspond that well with the computational estimates. And the purity mean was around 50%. So these are high variance and also moderate purity samples. First, we look for evidence of any SNV artifacts. This was after a little bit of refinement, looking at variants that are called across more than one SNV calling algorithm. So we use Mutect, and then it also has to be called in Strelka or multi-SNV, which uses multiple different samples from the same patient in order to call SNVs. And we found that there wasn't enough evidence to say that there was an enrichment of C to T transitions, which are customary. They occur a lot in FFPE tissue. So this is the 1 to 10% allele fraction. And then you can see that some of the samples do have quite a few more variants called than their fresh frozen counterparts. So the RARPs are the fresh frozen and the TX are the FFPE. So these are all post. However, we didn't find enough evidence to rule that out, that there are any, to say that there are any particular variant calling artifacts. And also the FFPE tissue happened to have on average higher purity, so it could have been because it was easier to call in the higher purity samples. So in order to determine whether we can use these samples for clonal analysis, we looked at the variant allele frequency of one particular sample that seemed to have quite high purity relative to the cohort. So this was the fresh frozen sample, and this is the FFPE. And then looking at the variants, so looking at what the correlation is between the variants for the variant allele frequency, it was pretty low at 0.36 Pearson correlation coefficient. So not great correlation, but we were wondering whether this is due to differences in the clonality, which they should be pretty much the same, or whether it's a difference in due to the variance of these samples. So simulating a few samples with beta binomial distribution allowed us to simulate a pretty similar Pearson correlation coefficient between taking the purity of both sample types, generating some wide variance distributions, and then looking at the correlation. So due to this, we thought that it was likely due to the high variance of the samples and the low coverage. So in order to test for whether you can track clonal changes in the pre and the post, with such noisy samples, we ran some simulations. So here was a clonal cluster between the two samples. It was the same proportion and the same thing with the other one. And running this through a clustering algorithm, this is dpclust, it's two-dimensional clustering. We can see that it's over-clustered quite significantly, so these are kind of the cluster means. However, you can still see that it's fairly symmetrical, and there's no kind of enrichment in any of the off-axis areas. So simulating with clonal change, 20% change to 80% from the TX to the RARP, you get a little bit more of an off-axis effect happening here when you do two-dimensional clustering. All right, so we decided to create this rough test for testing for clonal movement only in one direction, and if you've got high variance samples. So we create these kind of off-axis triangular components where we segment them and count the number of variants that fall into the CCF of all of these squares, and then also these ones as well. And then, so we've got the counts, and then we've got the counts for this one, and then we just apply a simple t-test to check for difference in mean, that the alternate hypothesis being that this is greater than this, and we can see that, yes, there is a shift from the pre to the post, suggesting that there is selection occurring. So in terms of the simulation, we ran multiple different variance parameters, so a very high variance, beta 1, and fairly high variance, beta 10. This was similar to what we saw in the neoadjuvant samples, and then decreasing the variance, and then trying to apply the test. So we applied it to all of the different simulated shifts, and found that, not much surprise, that this beta of 1 was too high to actually get any evidence for clonal selection. And then, so clonal shrinkage was 1 for everything because we were only testing in one direction. So we didn't expect any of these to be significant. However, we expected these to be significant, and they were. So these are changes at variance 10, 100, and 1,000. And you can see that it's not significant for no shift, which is exactly what we'd want. So you can see that we can use this test to in low variance samples in order to check for clonal changes. So now that we've got that, our simulations did not include copy number. So we correlated the copy number between the FFP and the fresh frozen. This is one sample. The purity is around 50%. And these are the copy number changes that are occurring. So these are chromosomes 1 to X, and this segment is the copy number that it thinks it is for the total, and then the minor as well. So looking at the FFP, you can see that the profiles are generally similar, but you can see that there's a lot more segmentation going on. So there's a lot more noise here, even though the purity is much higher. So the goodness of fit isn't as good as well. So this indicates that FP tissue, you've got some issues with SCNA calling. However, we found a pretty easy way to deal with it. So these are our ASCAT copy numbers. These are all the FFP samples in the post, and then this is the size of the copy number segment. So each segment is made up of a number of different frequencies at particular SNP locations. So the bigger your segment, the more accurate it should be, theoretically. And we found that this is the case with the FFP samples. So anything that was above this 1 megabase had pretty high concordance with the fresh frozen. So if the point is this sort of cyan color, it's concordant with the fresh. Otherwise, below this segment size, it was not concordant. And you can see that a lot of these aberrant copy number segments are very small. Battenberg, this is a subclonal copy number calling algorithm, and including that complication of subclonality, the concordance wasn't as high, but it was still higher above 1 megabase than below. So we decided to mainly use ASCAT copy numbers where we could, and do the cutoff in FFP samples, anything above 1 MB. So this is two-dimensional clustering of FFP in the fresh and the FFP sample. So what we would expect to occur is to have all of our clusters and variants being very close to this 45-degree line. However, you can see that that's not the case. You've got For instance, in the top left here in NAT09, this kind of looks like selection here and looks like selection there. However, when we looked at the estimate of the purity between NAT04, ASCAT and Battenberg, they were quite discordant. So these changes are likely due to the overestimation of the fresh frozen sample purity, and they were above 90%. I think they were about 96 or 97, which is very unlikely. You're very unlikely to have almost 100% pure samples. And the ASCAT purity estimate was maybe half that or sometimes a third. So these samples didn't have very good purity estimates. In fact, none of these samples had very good purity estimates in the RARP sample. So we couldn't really use these samples. However, you had clonal clusters appearing here, and mainly on-axis clusters, which was a good sign, and that means that the algorithm got the copy number calling right. And this one was kind of borderline. So applying this to the pre and the post, we ran this for any samples that we had, pre and post. And then, so there could be some evidence of selection occurring here. So if anything that's kind of off-axis, and if it's got the star, that means it passed that selection test, where I talked about the off-axis testing. So this one was apparently significant in this direction. However, it's possible that this is true selection. However, I'll talk about that a little bit later. These, we were pretty sure that they were poor purity estimates because we didn't really get that sort of clonal cluster occurring here. And there was also a lot of discordance between the ASCAT and the Battenberg algorithms. These ones also didn't have a clonal cluster appearing. This one did, that was fine, but it didn't pass the selection test. So we found some possible selection in some of these samples. So now that we've got a tool for a structural variant analysis in terms of clonality, can we use that on these samples to do some sanity testing or to check whether the same selection is occurring? So calling out SVs across this cohort, you've got the SV classifications here. So this is just a histogram, and the RARPs are the fresh frozen, and they're kind of ranked by number. And so you can see that the FFP ones are all kind of clustering on the left, and that's due to this very disproportionate amount of interchromosomal translocations. So this is the same with proportions that are being called. And these are very unlikely to be true. And you can see this by, if we just do a simple filter, more than one spanning read, this cuts down this proportion significantly, and it brings it more in line with what we see in the fresh frozen samples. So there didn't appear to be any problems with the SV calling for FFP, so that was good first step. Now let's see what the clonality looks like. So the variant allele frequencies actually had pretty good correlation. It was above 90%, which was great to see. And then when we ran it through SV clones pipeline, you can see that you also get very high correlation for the cancer cell fractions, which is very good. So this is the post fresh frozen versus FFP. So just sanity checking, everything should be pretty much on this axis. There was one sample that was significant for this clonal shift, which we know that there shouldn't be any clonal shifts occurring here. And that was because that kind of confirmed the poor purity estimate of the fresh frozen sample. But everything else looks very good, and we were able to use ASCAT copy number calling for this. So the purity estimate is a little bit better than Battenberg. Applying this to the pre and the post, some of the samples you can see that there's no real selection occurring. However, these samples had off-axis clusters. And so these are also likely poor purity estimates. NAT03 was one of them that was significant in the SNV clustering. However, here we don't really see any significance, so this is not corresponding with the SNV clustering results. And these samples are fairly similar to what we saw in the SNV clustering in that they're hampered by poor purity estimates. So You also notice that for NAT11, we've got the FFP and the fresh, so we can compare them directly. We've got two in the post, essentially. So in one of them, we had struggling to get the right purity. We can see that it's kind of been shifted off axis. However, in the TX, which is likely a lot better copy number purity estimate by ASCAT, they're kind of falling right on this 45-degree line, which indicates that there's no real selection occurring here. And the similar case with NAT13. So we were able to compare them. We've got a poor purity estimate, but looking at the FFP sample and comparing it to the other FFP, we can see that it's on axis. So there doesn't seem to be much evidence of clonal change occurring in these samples. However, this could be also something that is a clonal selection. However, we didn't see that in the SNV, so it would require some extra analysis. So looking at the pre and the post in the SV analysis running that, you can see that they're pretty much all on axis. So with the SV analysis, we're not actually getting any CCF changes occurring. In terms of the summary, so we used SV clone for this pan-cancer analysis. There was distinct SV and SNV heterogeneity patterns occurring across the different cancer types. We had this novel SCNR phenotype occurring, and then we also developed some strategies for for looking at changes in clonal shifts in these low-quality samples. And we found there was a little bit of evidence, but not much evidence for changes in the clonal dynamics. So acknowledgements, I'd like to thank all of my many supervisors, so Chris Hovens, Neil Corcoran, Jeff McIntyre, who's working at Cancer Research UK, and also Florian Markowitz, who was my host supervisor for a couple of months at Cancer Research UK, Cheng Soon Ong at Data61 and ANU, and Tony Pappenfuss here at WEHI. And the funding was provided by the Cybec Foundation, philanthropic PhD funding, and also the Endeavour Scholarship allowed me to visit Cambridge for a couple of months and do some of this project. So lots of thanks to them, and also we required a lot of compute facilities in order to perform some of these projects. So yeah, I'd also like to thank the PCOG working group and also my advisory panel, so Hongjian Zhu and Andrew Loney. And yeah, here's a list of some of the publications that have come out of this work. And one is in free review at the moment. So, are there any questions? Yes, Daniel?
What do you do with the soft clips that are longer than your 10 base pair cutoffs, but too short for your aligner to actually turn them into split reads?
Yeah, so
they don't count towards anything?
Yeah, they don't count towards anything. So we mark some reads as anomalous, and they are basically just ignored. Yeah. Yep, good.
Hi. You had a slide where you were looking at clones A and B, and you had two models where A and B occurred independently, or B occurred from A with a further development of A.
Yeah.
And you were saying that it would be hard to distinguish them without sort of earlier time points. If B is a further development from A, can't you look at sort of cumulative structural variants? Wouldn't B continue to have the original mutation of A and then a new one?
Yeah. There are some models that take a hierarchical approach, and you can do that. We didn't use a hierarchical approach with our algorithm, simply because you don't really have the sample numbers. So with SNVs, you've got a lot more. So you can fit them into your model hierarchically a lot better. But that's something that we chose not to implement because it would probably be a lot more difficult. There are some ways to figure out whether you've got this branching or this sequential phylogeny. And you can do that using pigeonhole principles. So if you've got two clones, one is 70% and one is 40%, so they're both subclones, well, you can't have a CCF higher than 100%. So one must give birth to the other clone, because you can't have both subclones, one being 70% independent and one being 40%. But if it's below that proportion, then it's more difficult, and you can do this via this stick breaking. So we've implemented a stick breaking algorithm, but there's also a hierarchical version of it. So this is like a Dirichlet process implementation. So if you do implement a hierarchical version, you can actually figure out lineages. There is a program that does it called PhyloWGS for SNVs, and a lot of the time you get very different phylogenies. So it's actually it's a pretty difficult problem, all in all. But yeah, you can get some idea.
No more questions, then we'll just thank Marek for the